these systems rely on salt water and an electrochemical cell. So you think, well, that's not very much, but the magic happens at the surface of those electrodes. The magic happens depending on the design of the electrochemical cell. And what you're doing is, in a very quantum world, is you're bombarding a whole ecosystem of chemicals, i.e. sodium, chlorine, hydrogen, and oxygen. You're bombarding it with electrons, and you're artificially saturating a solution with electrons in an electrochemical way. So you can produce solutions that are positively charged, solutions that are negatively charged. And understanding a little bit about that chemistry, you then understand why some of the solutions exhibit antimicrobial properties. And there are three things really which, sort of not going into the chemistry too much, but three basic things which really stand out. One is the pH of solutions. The second thing is the amount of active chlorine within those solutions. And the third thing is something that we call the oxidation reduction potential, or ORP. And that is the electrical quality of a solution. So the solutions that give rise to decontamination are positively charged. So what does that mean? That means they're really hungry for electrons. So whenever they stick onto something, the first thing they're going to do is nick or steal your electrons. Now, if you're a bacteria, or if you're a bacterium or a viral particle, that's bad news, because a lot of your internal workings rely on you being able to shuffle around these electrons. Yeah, that's your currency. And what that means is you can disrupt lots of really interesting binding sites on viral particles. As if you're a bacterium, it really disrupts your membrane wall, and it means that physiologically and biologically you cannot exist. You cannot function the same way. And that is how ECAS solution works. And ESOL is what we would call one of the top end of those solutions, because it provides solutions that inherently maintain a very high oxidative reduction potential. So they don't just rely on these chlorine species like many other decontaminants do. It also exerts an electron bombardment of a lot of the biology that we're trying to decontaminate. And that combination, if you like, of high electron bombardment or scavenging, combined with that high chlorine active species that are present in there as well, means that that's a really, really, really disruptive cocktail if you're a bacterium or if you're a viral particle. So then the natural extension to that is, well, why aren't these solutions toxic to humans? And of course, the reality is, in the bacteria world, in the viral world, that's a really, really small world. In humans, we're a bit bulkier, we're a bit heavier. You know, our structures and cell structures are a bit more robust. And one of the beauties about electrochemically activated solutions is that when they react, they do it really, really quickly. So you're talking about the speed of electrons, right? So that's really, really quick. And that's a benefit for us as humans. So, if you apply ESOL or ECAS to the skin, then the epidermis is quite a hardy organ. It's quite a robust structure. Then within 2 seconds, it's done. It's gone. So you don't have any lasting residual activity. If you want something which is quick, rapid, does the job in a very short time frame, leaving very little, if anything, behind, then ECAS becomes the perfect decontaminant. And of course, so decontaminating spaces, places where people frequent, makes it ideal, really.